So, hello students, welcome back to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master teacher, students. Good evening, good evening, good evening, each and every one of you students. Now, today is going to be the day one of your concept series. It's a small challenge, 4-day challenge, right? Today is going to be the day one, where in the morning we had a class from Diksha ma'am, and now I'll be taking a class on all the important concepts from your entire class 11th. You heard it right. So entire class 7 botany topics I'll be teaching you. So students, quickly let me know the chat right now, all of you, if I'm audible and visible to each and every one of you. Am I audible and visible, students? Quickly tell me in the chat if I'm audible and visible to each and every all of you. Karthik, Roshni, Supriya, Karthi, Vipul. Yes. Quickly let me know the chat, students. Quickly let me know the chat if I'm audible and visible to each and every one of you. Nice, nice. Now, let me ask all of you a very simple thing. Yes, good evening, good evening, good evening, all of you. Good evening. Let me ask all of you one simple question. Okay. How many times have you tried to study the chapter or the concept of kingdom fungi and you completely forgot after that? Like how many times it has happened to you that, you study the kingdom fungi, Ascomycetes, Phycomycetes, Basidiomycetes, and after exactly 1 day or 2 days or 1 week, you completely forget the entire kingdom. Has it ever happened to you, students? Has it ever happened to you? Students, quickly in the chat. Quickly in the chat, students. Has it ever happened to you that you're so ready and then boom, gone? Ever happened to you? Quickly in the chat, students, tell me. Kingdom fungi. Because I still remember when I was growing up, kingdom fungi used to be very tiny, but it is so confusing. Right? It is so confusing at times. So today's class, I'll make it the first concept for all of you is the kingdom fungi, and after today's class I'll give you the most amazing notes for it and you will never regret it. Okay, so starting off with the first topic for today, that is going to be your kingdom fungi, students. Quickly let me know in the chat, students, if you are ready. Fire emoji in the chat to make sure I know that yes sir, I am ready now. Nothing is going to stop me from learning the entire kingdom fungi. All of you, quickly in the chat, some fire in the chat, all of you. Now, first things first, all of you, first things first. Can anyone tell me in the chat which mode of nutrition is observed in your kingdom fungi? Quickly in the chat, students. Quickly in the chat. Which mode of nutrition is observed? In case of kingdom fungi, the mode of nutrition is heterotrophic mode of nutrition. What is the meaning of heterotrophic mode of nutrition? The meaning of heterotrophic is that all of these organisms in your kingdom fungi, they eat dead and decaying matter. They feed on dead and decaying matter. Okay, dead and decaying matter. Now, what is more interesting to understand here is, students, can anyone tell me, have you ever tried eating fungi? How many of you have tried eating fungi in your life? Because I have eaten. Yesterday night only I ate fungi. Anyone? Because I love eating mushrooms. If you're like me, mushroom biryani, so tasty. Okay, so all of us have eaten some way or another day, some way like fungi, right? Now I want everyone to understand here is that there are multiple types of kingdom fungi in kingdom, right? There is bread mold. You might have noticed, right? If you take one bread piece, okay, keep it in your in your house, put some water on it, you can grow fungi, free fungi for you. Okay, but don't eat this. There is also toadstools. There is your normal button mushrooms or Basidiomycetes member. Okay, there are something called as a wheat rust. It can be dangerous. Fungus can be used in yeast to make bread, to make alcohol, to make antibiotics. There are multiple applications of your fungi. But what is interesting to here note is that many students don't understand the difference between a mycelium and fruiting body. Tell me, students, how many of you know? How many of you actually know the difference between a fruiting body as well as, right, fruiting body as well as your mycelium? How many of you know? Let me tell you today. If you do not know the difference, let me teach you. Okay, listen to me. Now, if this is a fungus right here, do you see this entire lower part? This entire lower part is there, right? This entire lower part, right, that is called as vegetative part of the fungus. This is the vegetative part of the fungus. And let me tell you, students, vegetative part of the fungus, it is also called as It is also called as hyphae. It is also called as hyphae. So the vegetative part of a hyphae, vegetative part of a fungus is called as hyphae. What you are eating, no, Gungulum, that is called as the fruiting body. You are eating the fruiting body, not the hyphae. Okay. Now, small difference you understand. What is the difference between a hyphae and a mycelium? Understand the difference between a hyphae and a mycelium. Very simple difference. When there is a collection of hyphae, when many hyphae come together, that is called as mycelium. Is that clear to all of you? Group of hyphae, group of hyphae is called as mycelium. Mycelium. Very small difference, but if you don't understand, it becomes difficult to by heart all this. Okay, group of hyphae is called as mycelium. Okay. So now we have the mycelium growing underground. Now I'll show you one more image. I hope this will clear the entire confusion between what is a hyphae and what is a fruiting body. This is a typical mushroom. Here we can see under the soil, we can see the hyphae. Above the soil, we see the fruiting body, which we eat. Okay. Now, let's understand more about hyphae. Okay. Now, there is something special type of hyphae. For example, students, some of the hyphae will have something called as cross wall. Cross walls. Do all of you see the cross walls? Here. They are called as cross walls. Cross walls. Okay. Now, understand. What if there is no cross wall? Do you see, students? There is no cross wall formation. In such condition, it becomes multi-nucleated. When there are no cross formation, it becomes multi-nucleated condition, and such a condition is also called as what? Cenocytic hyphae. So multi-nucleated condition hyphae is called as cenocytic hyphae. Is that clear to all of you? Students, all of these are terminologies which can be asked as such directly. Cenocytic hyphae is called as what? Multi-nucleated hyphae. There is no cross wall formation. No cross wall formation. Clear all of you? Clear. Now students, let me teach you one more thing here. Your normal septate hyphae. It's also called as septate hyphae. Septate. Septate means cross walls. Septate hyphae can be of two types. Can anyone tell me in the chat? What is called as septate hyphae? Any two types of? For example, understand students. If this is a hyphae, here I have a cross wall formation. Here I have a hyphae, and here also I have cross wall formation is there. Okay. Understand all of you. Understand. In the first type of septate hyphae, I will find only a single nucleus. In the second type, I'll find one small, one large nucleus. Two nucleuses per cell here. Two nucleus. Such a type of hyphae, students, where there is a single nucleus, it is called as monokaryotic. Monokaryotic hyphae. This structure is called as what, students? Dikaryotic hyphae. Di means what, students? Di means two. Di means two. So dikaryotic hyphae means two nuclei are there in this hyphae. Clear all of you? Clear, students, all of you? Everyone in the chat quickly. Can I see some fire emoji in the chat to know that it's clear to all of you? Okay. So monokaryotic as well as your dikaryotic hyphae. They are a type of branched hyphae. Now students, reproduction. Reproduction in the case of fungi can be three types. Vegetative, asexual, sexual. What you need to focus mainly is asexual, only the spores. And main point here is going to be sexual reproduction. Okay. Now listen to me, all of you. Vegetative can be in the form of budding, which you have seen in yeast. Okay. It can be in form of fission can be there. What is fission? One cell dividing into two cells. One parent cell dividing into two daughter cells. Then there is fragmentation. Now, then there is something called as asexual reproduction. In asexual reproduction, there are mainly something called as asexual spores. Asexual spores. Now, students, I'll give you a table. It becomes so easy to understand, and I'll move aside for all of you. Can all of you see the table, students? This is a table right here for all the asexual spores given in your syllabus. You will never forget them. Now, for example, there is something called a zoospore, sporangiospore, and conidia. What is the difference? Zoospores are motile. Sporangiospores are called as aplanospores, which are non-motile, and these are also called as aplanospores, which are again non-motile. Students, download these notes. You can get them on my Telegram channel, Basavaraj Sir Biology. Very easy. Okay. Now, they are called as endogenous spores. What is the meaning of endogenous? Tell me, students, what is the meaning of endo? Endo means inside. Endogenous spores are produced inside the body. Okay, endogenous, exogenous. Let me show you a small diagram. You'll understand better. Okay, for exogenous spores, better clarity I'll give you. Students, can all of you see the spores here? Can all of you see the spores? See, so sporangiospores are present inside a body here. Can all of you notice this? This is sporangiospore inside the sporangium. Inside the sporangium. Well, if you look at a conidia, conidia is an example of exogenous spore. Can all of you see? It is present separately outside. Exogenous spore. Exogenous spore. Okay. So conidia is going to be exogenous spore. Exogenous. Okay. Now, mainly mode of where is it found? In water. Transportation by water. Terrestrial, air and water. Now, which of the following classes do you find? This one is found in your Phycomycetes. This one is found in your Rhizopus and Mucor, and Conidia is found in your Ascomycetes and Deuteromycetes. Now, some of you will be asking me, sir, Phycomycetes is here, Asco is here, Deutero is here. Asco and Deutero, Conidia, Rhizopus and Mucor, Sporangiospore, Zoospore is going to be Phycomycetes. Now, some of you will be asking me, sir, where is Basidiomycetes? Can anyone tell me in the chat, students, which type of spore, asexual spore, we find in the case of Basidiomycetes? Anyone? Anyone who can tell me, students? If you know, tell the answer in the chat. Which type of spore you will get in Basidiomycetes? Anyone? Which type of spore do you get in Basidiomycetes? Quickly, students, quickly. All of you. Students, don't worry, you will get all of these notes. You will get all of these notes. Anyone who can tell me which type of spore do you get in Basidiomycetes? Quickly, students, quickly. I'll give you exactly, students, exactly. So please remember that, students, in the case of Basidiomycetes, asexual spores are absent. First point we need to remember, PYQ, asexual spores are absent here. So please remember this point. Don't worry, I'll teach you again one more time. This one will come up. Okay. Now I'll show you a table, students. Now I'll show you sexual reproduction. Sexual reproduction, see, vegetative done, asexual done. Asexual, you learned about 3 types of spores. Okay. Now under sexual reproduction, it is special here. Sexual reproduction, there are 3 main steps which you need to remember. First step is called as your plasmogamy. Plasmo means your protoplasm. So in plasmogamy, fusion of protoplasm is happening. Okay. Then protoplasm will fuse here. Karyogamy. In karyogamy, there is a fusion of nuclei. Once there is a fusion of nuclei, that will result in the zygote formation eventually. Once the zygote is formed, it undergoes meiosis to produce spores and plant body we get. I'll repeat again. Plasmogamy, karyogamy, zygote formation. After zygote formation, meiosis, very important, spores, plant body. I will tell you an extra point here, students. Every single sexual spore is always produced by meiosis, while your asexual spores are produced by mitosis. Okay. Now, sometimes what happens is between plasmogamy and karyogamy, there is an additional stage. The additional stage is called as what? Now listen to me. This is a very important statement here. Sometimes what happens, students, in certain fungi, protoplasm will fuse, but nucleus will not fuse. What is you telling, students? What are you telling me? Protoplasm is fusing, but the nucleus is not fusing. Have you seen this? Particular phenomena anywhere, students? A particular phenomena where In a mycelium, the protoplasm is fusing, but the two nuclei are present as such. This stage right here, where there are two nuclei per cell, that is called as dikaryophase. Dikaryophase. Where within one cell we can find two nuclei because nuclei are not fusing, and that is called as N plus N condition, dikaryon or dikaryophase. It is observed in Ascomycetes and Basidiomycetes. The most important point here, students. The most important point. So I'll repeat again, understand. Students. Protoplasm is fusing, but the nuclei remains free, and they are called as dikaryon phase. Dikaryon phase. Now, we have 1, 2, 3, 4. 4 different classes under kingdom fungi. 4 different classes. Now, is it very difficult to remember it? Yes, it is difficult. There's a very high chance that you might forget if you write it in a paragraph wise. So to overcome that situation, I have made a table for all of you, and if you refer this table, you will never lose marks. Let me show you. See this. This is a table right here, students. This is a table right here. I hope it is visible to all of you. I hope it is visible to all of you, students. This is a table right here. Okay. Let me read it once for you. I'll give you the screen. I'll give you the entire notes, so you don't have to worry about anything here. Okay. Now, first, characters here. Phycomycetes, Ascomycetes. So, characters. Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. All 4 are written here. Okay. Now, what is the common name? Common name is called as algal fungi, sac fungi, club fungi, and fungi imperfecta. I'll tell you why it is called as fungi imperfecta later on. Okay. Mycelium is cenocytic. I told you. What is the meaning of cenocytic? Multinucleated in nature. Multinucleated in nature. Aseptate. Septate, septate, septate. Habitat. Decaying wood and obligate parasite on plants. Terrestrial. That is coprophilous. The meaning of coprophilous is it is found in your dung. That is waste, cow dung and everything. Okay, dung. And most of them are decomposers. Basidiomycetes, tree stumps, wooden logs, and parasitic in nature. Minerals cycling by the decomposing. So main function of Deuteromycetes is mineral recycling. Spores, asexual spores. Sporangiospore, conidia. I told you, Basidiomycetes no spores. Here we get conidia again. Conidia, conidia, sporangiospore. Beautiful, beautiful. You will never forget all this. Next, sexual spores. Here we find zoospore, ascospore, basidiospore. See, this is very easy to remember. Very easy to remember. Ascomycetes, ascospore. Basidiomycetes, basidiospore. But in the case of Deuteromycetes, you will not find any types of sexual spore. In fact, sexual reproduction is completely absent here. It is completely absent here. Completely absent. Sexual reproduction is completely absent here. Now, apart from the students, apart from that, do we find sex organs here? Yes. Yes. No. No. But you might ask me, sir, see here it makes sense. No reproduction, no sexual reproduction, no sex organ makes sense. But what about Basidiomycetes, students? In the case of Basidiomycetes, we have sexual spores, but no sex organs. This here is where they catch you. This is where they catch you. There are no sex organs in the case of your Basidiomycetes, but there is sexual reproduction. How? I'll teach you that also today. I'll teach you that also. No. Last one. Fruiting body? No fruiting body. Here we find ascocarp, basidiocarp. Ascocarp will give you ascus or asci. Basidiocarp will give you basidium. Now, what is basidium? What is basidiocarp? Now I will teach you in the next 2 minutes. All of you ready, students? All of you ready now? Students, how are these notes? Tell me first of all in the chat. This, if you just remember this one slide, 2 slides, your entire, all 4 classes, most important points is written here only. You don't even have to forget about, you don't have to even put an effort after this. Just read these notes, download these notes in the Telegram channel, we'll give you a Telegram, right? And just read the NCERT one more time, you will never forget kingdom fungi. Okay, I would always recommend all of you to write this on your own also. Okay, write this on your own. Now, understand what exactly happens in your Ascomycetes and what exactly happens in your Basidiomycetes. First, we'll focus on Ascomycetes. Understand. In the case of Ascomycetes, sexual reproduction is by conjugation in yeast. Gametangial contact can also be found here. Now, what exactly happens here? Understand. Here we have two sex organs. I told you, Ascomycetes, sex organs are present. They are present. Okay. Dichorogamy is happening here. Dichorogamy. Understand what is dichorogamy? Understand. First of all, there is something called as antheridium here. Antheridium. And ascogonium. Don't write archegonium here. Archegonium is in plant kingdom. Okay. So ascogonium. What will happen? First episode, students. First step is plasmogamy. Second is karyogamy. Third is zygote formation. All of you remember that. So plasmogamy happens here. Now, in the case of Ascomycetes, after plasmogamy, we do not get karyogamy. In between, there's a stage. I told you, students. I told you, in the case of Ascomycetes, after plasmogamy, karyogamy does not happen. There is an intermediate stage that is called as dichorophase. That is what is happening here. We have dichorophase happening here. Dichoro. N plus N condition, two nuclei. Now what happens, understand. On this particular mycelium, there will be development of ascus. This is called as ascus or asci. It is a structure. This is a structure which has the two different nuclei. Now remember, students, can anyone remind me, what do you call Ascomycetes as? Which type of fungi? We call them sac fungi. Here is the reason. Students. Here is the reason. Sac shape. Sac fungi. Okay. Now what happens? Understand. The two nuclei will fuse together. Zygote formation. So plasmogamy over, karyogamy over, zygote formation over. Now once there is zygote formation, it undergoes meiosis to produce ascospores. So what is ascus? Ascus is this entire structure. Ascus is the sac-like structure. Ascospores are the spores present inside the sexual spores. Now, you might be wondering, sir, what is ascocarp then? You told me this entire structure is called as ascus. Spores are called as ascospores. Then what is the ascocarp? Understand. What is the ascocarp? All of you see this? This is called as ascus. One entire thing is called as ascus. It has ascospores on the inside. When multiple ascus, can you see? Multiple of them ascus will come together. The entire structure is called as ascocarp. So ascocarp is a fruiting body. Ascocarp is a fruiting body which has many what? It has many ascus here. Multiple of them, 1, 2, 3, 4, 5, they'll be arranged together. The entire structure is called as your ascocarp. So understand what is ascus, what is going to be ascospore, and what is ascocarp. Three different things. We have the ascocarp here. Ascocarp. Done, students? Done? Beautiful. Now same way you need to understand what exactly is happening in Basidiomycetes. I told you in the case of Basidiomycetes, there is no sex organ. What happens here? In the case of Basidiomycetes, there is something called as your plus and minus mycelium. So we have your two types of primary myceliums here. Okay. Vegetative. Both the vegetative mycelium will fuse here. Okay. So what is happening here? Plasmogamy is happening. Then we get a dikaryon. Then we have a club-shaped structure called as club fungi, right? Club-shaped structure here, which has both the nuclei. They will fuse again to form zygote, meiosis, and we find basidiospores here. Can all of you see basidiospores? Basidiospores. And if you remember, students, I told you here, basidiospores, right? Basidiospores are produced. Basidiospores, ascospores, right? The key difference here is that ascospores are endogenous, while your basidiospores are exogenous. Can all of you see exogenous? Now all of you tell me, this is called as basidiospores. Okay, basidiospores. This structure is called as basidiocarp. Okay. One second, students. Basidium. Then we have basidium here. Okay, one second. First of all, we have basidiospores. This is basidium. Basidium. Okay. When multiple basidium join together, what do we get? When multiple basidiums join together, we get the basidiocarp, that is the fruiting body. Fruiting body. That is going to be basidiocarp. That is how the reproduction happens in the case of your Ascomycetes and Basidiomycetes. Okay, so let us soak in. Here is your basidiocarp. Can all of you see the club-shaped structures? Club-shaped structures, multiple basidium together. And you can see spores present exogenously here, exogenously. When multiple basidium join together, they make up the entire fruiting body. That is called as basidiocarp. So basidiocarp is the fruiting body, basidium is a structure, basidiospores is the spores. Here, ascocarp is the fruiting body. It has the ascus is the structure which has the ascospores, and that is the end of your entire kingdom fungi. Phycomycetes, nothing to remember. What I told you, you should remember. In the case of your Basidiomycetes, done. Deuteromycetes, fungi imperfecta, that's all. Apart from this, the only thing which you should remember is the examples, and you're done, students. And you're done. That's it. How was it, students? How was it? Did all of you get a better perspective of entire kingdom fungi, students? Did all of you get a better perspective? All of you. How was it? Better perspective? So how does the reproduction happen? Because you might not have learned this way everywhere. All of you. Understood or not? Tell me. Now I will teach you one more concept which is the most difficult concept in your entire class 7th actually. If you just see NCERT, you will never understand. If you just, next topic which I'll teach you is the topic if you read NCERT, you will never understand. That is my guarantee. That is my guarantee. I will teach you such a concept now. Ready all of you? Ready now? All of you? Smash the like button. Smash the like button, all of you. Can we start, all of you? Can we start? Students, I want to understand where is everyone from? All of you are from where? Which location? I want to know. Because I know most of our students are from South India, obviously, and Northeast. But just asking one more time. Just asking one more time. Just to talk to all of you. I need a reason to talk to all of you, no? Where is everyone from? Yes, Mohan, Shivu, Supriya, Meghana. Meghana, I know. Meghana from... Yes. I have a lot of topics. Students, it depends on how much you want to learn. I have a lot of topics. I can go on for 4, 5, 6, 7 hours. I have so many topics. Okay. Don't underestimate. Bangalore, yes. Now, next topic coming up. All of you ready? Okay. Now, students, let me challenge all of you. Let me challenge all of you. If I show you this structure, what do you think it is? If I show you this structure right now, what structure do you think it is, students? What mental image does it create? What type of mental image do you create when you see this structure of your... I'll not say the answer. Anyone? Because I'll teach you right now the way which we will clearly understand the concept, not just by heart the stupid thing which is given in NCERT. Anyone who can tell me? See, we always say this is flagella or cilia structure, but it doesn't look like flagella, no? Does it look like cilia and flagella? It does not. So let me teach you today, students, what exactly are we dealing right now here. Ready, all of you? Ready? See this. Students, does this look like cilia and now does this look like a flagella? All of you tell me, does this look like a flagella now? Flagella. In eukaryotes, all of you know, flagella is a structure which helps in motility, or cilia also, as a matter of fact. Does it look like something which will help you in motility now? It does. It does. Okay. Now, what we need to focus here is the cross section now. This is a cilia and flagella. This is a cilia and flagella. This entire thing is the flagella, which is made up of a cytoskeleton. The entire flagella is made up of a cytoskeleton called as what? Anyone? What is it called as? It is made up of a cytoskeleton called as microtubules. Microtubules is a type of cytoskeleton. What is cytoskeleton? Understand? Like, for example, our body is made up of bones. Our body is made up of bones, right? It has bones in it. Why? Bones helps in the framework. It gives a framework. Cytoskeleton is also a type of protein structures present inside the cell, which provides a framework to the cell. Like how we have bones, inside the cell we have cytoskeleton. One of the examples of cytoskeleton is microtubule. Okay. Now this entire thing is called as made up of microtubule. Now, in this entire microtubule, students, if you see a cross section from the top view, what do you see, students? From the top view, if you see, you will see a structure like this. Something like this. Now, students, you tell me in the chat, does this resemble what is given your NCERT now? Does it look like what is given you NCERT? It does. It does. So, what they have given you in your NCERT, students, without actually knowing the structure of it, it is very difficult to understand. The structure which they have given you in NCERT is the cross section, top view. Now, how many of you understood? Tell me in the chat quickly. How many of you understood what we're dealing with here now? How many of you understood now? Tell me in the chat quickly. Now, this diagram makes sense. See, I don't even know why they've given you electron diagram, electron microscopic diagram. It doesn't make sense. Instead, they could have given a flagella diagram and show the top view for you to understanding, right? So that's why I'm here for all of you. See this. This is the structure, top view, and you see the top view in your NCERT. Okay. Now, students, before we go to cilia and flagella, let me tell you about cytoskeleton. There are three types of cytoskeletons: microtubules, microfilaments, intermediate filaments. What we'll be studying here is the microtubules, which are mostly hollow, unbranched, non-contractile. Understand now, students. Understand. What is cilia and flagella? Understand. Cilia and flagella is a hair-like structure whose main function is motility. Okay. Main function is motility. And they emerge from the basal body. Now, your NCERT says a line. It is given your NCERT. They emerge from basal body, but what is a basal body? What is a basal body? Understand again. This structure which you saw here, students, this structure which you saw here, from where the entire flagella is coming out, this entire structure is called as basal body. Understood? So this is the concept which no one teaches you, right? This is called as the basal body. From this basal body, the entire flagella is coming out. From the basal body. Understand? Okay. Now, there are two types, right? I told you. Cilia and flagella, difference you understand. Cilia is shorter, small. Flagella is longer. It has oar-like movement. Understand what is oar-like movement? Understand. Students, tell me, how many of you traveled in boats? How many of you traveled in boat like this in your childhood? Tell me, students. Tell me, how many of you have traveled in boat like this? Tell me. If you have traveled in this boat, what do you need to do? Hold the two things and move it. Move it like this or push it back. Now, when you push, right, will the boat go front? Yes. Will the water move back? Yes. Same logic is applied in the movement of cilia. Cilia is a tiny hair-like structure. It will move the entire liquid media or aqueous media around it, and the entire structure will move front. Just try to imagine it. There is a small cilia here. There is small cilia everywhere throughout the body. And let's say I have liquid everywhere. Now what is happening is, students, this entire cilia is pushing the water and it is moving front. And this entire thing is present throughout the body, and we have a flagella at the end. That is called as oar-like movement. Okay, throughout the body, present at the end. Now, students, understand this diagram. Okay, we will understand this diagram with the help of my diagram. Okay, listen to me very carefully, all of you. 2 minutes. Give me 2 minutes. Just 2 minutes. Okay. I told you this entire structure of your cilia and flagella, it is made up of microtubules. Yes or no? Yes, sir. Microtubules. Now here you need to understand here is that in the periphery, there is microtubules. How many? Let's count, students. 1, 2, 3, 4, 5, 6, 7, 8, 9. We have in total 9 periphery microtubules which are present as doublet. 9 periphery present as doublet. What is the meaning of doublet? 1 ring, 2 ring. Doublet. Doublet. Understand now. In the center also we have microtubule that is called as central microtubule, which is joined by a bridge. Joined by a bridge. Central, how many are there? 2 are there. So 9 plus 2 arrangement. 9 plus 2 arrangement. Now, this pink color region is called a central sheath. This central sheath is attached to the peripheral microtubule. In the middle, this small thing you see, which is linking both of them, that is called as a linker. That is called as a linker. Linker. Okay. One second. Linker is here, students. No, sorry, forgot. This is linker here. Between the two doublets, linker here. This line here is called as radial spoke. This line here is called as radial spoke. Line here is called as radial spoke. Okay. Then central sheath and linker is present between the two green things. Linker is there. Okay. Now, see this diagram. Here we can see 9 perimicrotubules, central 2, middle bridge. We can see here radial spokes. Where is radial spokes present here coming out. Radial spokes. So this is the structure of your cilia and flagella. Done. All of you. Now, students, do you want to learn the structure of centrosome or not? You tell me in the chat now. Do you want to learn the structure of centrosome or should I skip it? All of you in the chat. Do you want to learn? Again, it is not given in NCERT. Again, if you read NCERT, you will not understand the structure of your centrosome because it is that advanced concept which they don't give you sometimes. Do you want to learn? All of you in the chat? Let me look at the chat. Let me look at the chat right now. Students, you have not even liked the video. I thought it will be 200 likes at least. Students, smash the like button quickly. Do you want to learn or not? All of you. Or should I go to the next topic? I have many topics today. Want to learn, right? Okay. Understand. Now, first of all, students, tell me in the chat, which are the two organelles, right, present in a eukaryotic cell, which do not have any membrane? Because we generally tell prokaryotes are non-membrane bound, eukaryote are membrane bound. But there are two organelles, there are two exceptions under eukaryote which does not have any membrane. The first obvious answer is ribosomes, which does not have any membrane, right? The second answer is going to be your centrosome, right? Centrosome is present exclusively to your eukaryote, right? As well as exclusively present only in your animal cell. It is completely absent in the plant cell. Again, all of these are PYQ, students. I am not just teaching you one concept, I am teaching you so many concepts which are interlinked. So centrosome is a organelle which is exclusively only present in your animal cell. Now understand the structure. It is non-membrane bound. Okay. It duplicates in the S phase of your cell cycle. Okay. If this is your entire structure is called as centrosome. This entire structure is called as centrosome. One centrosome is made up of two centrioles. See, this entire thing is called as what? Entire thing is called as centrosome. One centrosome is made up of two perpendicular structures called as centrioles. Let me show you all of you. Let me show you. Is everyone able to see this diagram? Is everyone able to see this? This right here are the two centrioles which are perpendicular to each other. Can all of you see? They are perpendicular to each other. These are the two centrioles present inside centrosome. Inside the centrosome. Two centrioles. Now let me ask all of you a very simple question. Very simple question. Okay. If you look at the centrioles, students, centrioles, are they hollow or are they solid? Tell me, students. Quickly in the chat. Students, are these structures hollow or are they solid? You will tell me, sir, this structure right here, students, is hollow in nature. Now, if this structure is hollow in nature, can anyone remind me which type of microtubule is present here? This each one, which type of cytoskeleton? I told the answer by mistake, students. Answer by mistake. Okay. So, entire structure is made up of microtubules again in your centrioles. So all of you can see the arrangement here. Can all of you see the arrangement? Can we count? 1, 2, 1, 2, 3, 4, 5, 6, 7, 8, 9. There are again 9 periphery microtubules, but again they are triploid in nature. They are triplicates here, not triploid, triplicates here. Okay, and in the center, students, in the center, do you find any type of microtubule here? Can anyone tell me? In the center, do you find any type of microtubule here, students? In the center, tell me in the chat quickly. Triplet? Absolutely not. So in the center here, I do not find anything. In the center here, students, I do not find anything here in the center. Okay. So before we go to the structure, one more point is missing. This entire thing is called a centrosome, two centriole perpendicular to each other. This entire structure is covered by a small membrane-like structure, not membrane-like structure called as amorphous pericentriolar or kinoplasm or centrosphere. Centrosphere, kinoplasm, amorphous pericentriolar space. So it's a membrane-like structure, very fine structure around it. Okay. Now here the structure right here, students. Again, here. Now if you can see, students, it has again 9 periphery microtubules. 9 are there. But in the right, in the center, there is nothing but a protein layer called as hub. So in the center, there is 0 microtubules. Here we have hub here. So 0 microtubules in the center. So the arrangement in the case of centrioles, in the case of one centriole is 9 plus 0 arrangement. Cilia and flagella was 9 plus 2 arrangement. Okay. Here again we have radial spokes coming out. Okay. Now, if you remember the structure of your flagella, I told you this is the flagella, this is the basal body. But did you know that the basal body actually has 9 plus 0 similar to centriole, and the upper part is 9 plus 2. How many of you knew that, students? How many of you know that? 9 plus 2 flagella, basal body is also 9 plus 0. 9 plus 0. So students, what is the main function of your centrioles or centrosome? In fact, centrosome, the main function is in cell division. What do? They radiate spindle fiber. For example, during cell division, they will radiate out the spindle fibers. That is the function of your centrosome. And with that, students, we come to the end of one more topic. Done, students? How was it? How many of you understood? Students? Tell me honestly, because it is a concept. If you just read the lines of NCERT, there is a 99% chance you will not understand, because it doesn't give you clarity, right? After the visual representation, all of you might have understood the entire concept. Tell me yes or no. All of you in the chat. Students, smash your like button. I want to see 200 likes very soon. What is this? Let me tell you again, no one will teach you such animation wise, no one will give you such clear cut information. All of you. Next topic, students. Next topic now. Can you go to the next topic now, students? One small connecting topic which everyone should know, right? How to manage KCET and NEET, very difficult task. But if you read for NEET, KCET is going to be easy for you. But in KCET, please focus on mathematics separately. That's all, bro. Rajesh, that's all. Okay. Now, Supriya, keep a track of my likes. Now, students, this is a structure of your entire structure is called as chromosome. One chromosome has 2 sister chromatids. Okay. Now, students, tell me in the chat quickly. If I make a chromosome like this separately next to it, what do I call this one chromatid and this chromatid? Will I call them again sisters? Tell me yes or no in the chat. All of you. Will I call them sister again? Will I call this chromatid and this chromatid sister? No. They are non-sisters. They are going to be non-sister chromatid. Non-sister. They are going to be non-sister. Okay? Non-sister. Done. Now here the middle part is called as a primary constriction, also called as centromere. Centromere. Around the centromere there is a protein cup-like structure called as kinetochore. All the spindle fibers will always attach to the kinetochore. At the end, we have a secondary constriction called a satellite. Below the satellite, the region is called as a telomere. Telomere. This much you should know before we go to the next topic. Now, if I ask all of you, which is the most difficult concept in your entire cell cycle and cell division, what will you tell me, students? Tell me in the chat. Yes, target reached down. Thank you so much. Arigato gozaimasu, all of you. Now, next target is 300 likes. Let's go. Okay. Now, if I ask you what is the most difficult topic in your entire cell cycle chapter, you will tell me, sir, I can understand mitosis. I can understand the cell cycle. Meiosis is the difficult part. Meiosis is the difficult part where I usually find difficulty in understanding the first step in meiosis. That is your entire structure, entire process of prophase I. What is leptotene, zygotene, pacotene, diplotin, diakinesis? How many of you find it difficult? Tell me in the chat, quickly in the chat, students. How many of you find it difficult to understand and remember leptotene, zygotene, pacotene, diplotin? What exactly happens in every single step there? How many of you understand? Difficult. Especially prophase I. You don't lie to me. Be honest. Be open, because I will teach you that part. Only that part today. Because this is a concept where we increase the marks, not be in the same level. If you're coming to my class, there is a very high % chance that you'll become a better person before you go. When I say better person, knowledge wise. Okay. Nucleolus and nucleoplasm. Nucleoplasm is the cytoplasm of your nucleus, and your nucleolus is the structure which mainly produces rRNA. Okay. Can we start, students? Can we start, all of you, quickly in the chat? Can I get some confirmation in the chat? Can I get some confirmation? Just say ready, sir. I will start now, students. And all of you, did you share the video? Students, again I'm telling you, this is my Telegram which you can join, where I'll be sending the notes. Basavaraj Sir Biology is my Telegram where I'll be sending the notes for all of you. So join it. Okay. Numerical problems we'll do separate session for numerical. What is there to worry? Okay. Can we start, all of you? Yes. Now, first of all, all of you need to understand what is meiosis and what exactly is happening in meiosis and where does meiosis takes place. Okay. Now, what exactly is happening in meiosis? Understand first of all. What exactly is happening in meiosis? There is a change in chromosome number. For example, in meiosis, if a particular cell is diploid, if it undergoes meiosis, it will become triploid, quadruploid, penta, or haploid. It will become haploid in nature. There is a reduction by half in the number of chromosomes. It is also called as reduction division. Why? Because there is a reduction in chromosome number. Okay. Now, understand, all of you. In the entire meiosis, it is divided broadly into two categories: interphase and M phase. Interphase has G1, S, G2. All of you are aware. Can anyone tell me in which phase of your interphase DNA duplication happens? Anyone? Quickly, students. Quickly. In which phase does the DNA duplication happens in your, is it in G1, S, G2? In the chat, all of you. Okay. Now we come to entire M phase, also called as meiosis. Here meiosis I, meiosis II, two different types of meiosis there. There's a major difference. Meiosis I has four main stages called as PMAT, PMAT, mat. You sleep on this mat. Okay, PMAT. That is prophase, metaphase, anaphase, telophase. Okay, done. This entire meiosis I is called as your reduction division because the ploidy is changing here. While your meiosis II is called as equational division. Why? Because meiosis II is very similar to your mitosis. Brother, sister, they are almost twins. Let's call them twins only. Okay. So meiosis II, I'll not be teaching you today. Very easy. If you know mitosis, you know meiosis II. What I'll be focusing on is, especially today, is entire meiosis I. And all of you need to remember that between meiosis I and meiosis II, in the middle there is one more stage called as interkinase. Interkinase is very similar to your interphase, but small difference. The small difference is that in your interkinase, there is no DNA replication. No. In S phase, there is no DNA replication here. Done? Okay. Now let's understand meiosis I. The entire meiosis I is divided into PMAT, all of you know. PMAT. Prophase, metaphase, anaphase, and telophase. Or telophase. Some of you say it. Okay. Now understand. Here, this entire prophase itself is again divided into multiple stages. Prophase. Here is 1, 2, 3, 4, 5. So entire prophase divided into how many? 5 different phases again. Only prophase divided. Okay. Now, it is divided into leptotene, zygotene, pachytene, diploidine, diakinases. Is it difficult to remember? It might be for some people. So there is a small trick here. Let zebra play during the day, L4 leptotene, Z4 zygotene, P4 pachytene, D4 diploidine, and D4 diakinases. Done. Now let's understand every stage one by one. I want everyone to focus very well. Do not look here, there, nothing. Just listen to what I'm saying, understand the diagram, and it will be stuck in your head. Okay, let's start. First one is called as leptotene. What exactly happens? I'll tell you. All you need to do is remember 1 or 2 points. That's it here. Leptotene. First point in your entire prophase 1. Chromosomes gradually becomes visible under light microscope. Second point. So chromosomes which are in the form of chromatin, they become gradually visible under light microscope now. Visible. That's all. 2 points all of you can remember. Compaction of chromosome continues. That's all. So what is happening in the leptotene is going to be condensation of chromosome is starting. Chromosomes are becoming gradually visible under light microscope. That's it. 2 points done. All of you. Now, next point. Next is called as zygote. Zygote is the second stage. All of you know that. Now, what exactly is happening here? Understand. Chromosomes start pairing together called as synapses. Understand. Students, when you were in class 6th, 7th, 8th, right, there used to be a group activity. The teacher used to tell you, go on, run and pair with someone in your class to form a group. Now, what do you used to do? Do you make a pair with your best friend or do you make a pair with some random person in your class? Tell me in the chat, all of you. Do you make a pair with any random person in your class, or we go with your best friend? You're like, bro, listen, both of us will do the project. You are my partner now, right? Does it happen? Yes, it happens. Same concept is applying here. In zygote, what is happening here is 2 chromosomes are pairing up here, and the process of pairing up is called as synapses. And these chromosomes are called as homologous chromosomes, which are because they are alike. They look like each other. Okay, they are best friends. Here. The process of coming together is called as what? Synapses. So chromosomes start pairing is called as synapses. That is the first point which you need to remember here. First point. Now understand second point. So 2 chromosomes have come together now. Now between the 2 chromosomes, right, there is a complex formation here. It happens to you also, no? Imagine you, both of you friends became one partner here. Okay. Will you still fight? Yes, you will still fight. Why? Because your idea is different, your friend's idea is different. You're like, you keep fighting with each other. Same thing is happening here. There is a complex formation between the 2 different chromosomes here. Can all of you see? Complex. And this complex is called as synaptonemal complex. So here, second point is going to be formation of synaptonemal complex. First point is what? First point is synapses coming together. Between complex formation, that is called as synaptonemal complex formation. Done. Now, third point. This entire stage is called as bivalent or tetrad. Now, you might ask me, sir, is bivalent and tetrad same to same? Yes, it is same to same here. Only the difference is with which respect you are looking at it. Understand? It is called as bivalent because Two chromosomes are here. 1, 2. Two chromosomes. Bivalent. Bi means 2. It is also called as tetrad. It is called a tetrad because how many chromatids we have? Chromatids. 1, 2, 3, 4. 4 chromatids, tetrad in nature. Tetrad. Both meaning is same. The way you're looking at it is different. Understand? Like how you are one person only, but in your school you are jolly. In your school you'll be doing all kinds of nonsense in school. But as soon as you come home, dad is coming to the house. What will you do? Nama you'll put Pitashri, come to your house. That's how you're like full decent. You become decent. So both the same person only. Both the same person. The only difference is how you're looking at it. Okay. So it is called as tetrad or bivalent. That is the third point here. Okay. Zygotine. Understood, students? Zygotine here. So first is called for zygotine. First point is synapsis. Second point is synaptonemal complex. Third point is bivalent or tetrad. Bivalent or tetrad. Done. Let's go to the next thing. 3 points done here. Next is called as packaging. Next level. What is packaging, students? Packaging is very simple. You're doing some exchange offer. It happens in school also, right? You will get one tiffin. Your special friend will bring one more tiffin. What do? You're like, my mother has made this. You take this. One piece you take. Your special friend will tell, my mother has made this. You take one piece from here. Okay. Who knows? You would have only made it. You will tell your mother has made it. Okay. You're doing exchange offer there. Exchange offer. Right. Exchange offer. Right. Now, that exchange offer is happening in your packaging. Yeah, packaging. Okay. Understand all of you. Sir, can you explain formation? See, you don't have the formation of synaptonemal complex. Just remember that between two synapses, we have synaptonemal complex formation. That's all you remember. Okay. Now sharing is caring is happening here. Understand what is sharing is caring here. Okay. First of all, the first point here is that bivalent chromosomes clearly appear as tetrad here. Now, if I ask you a question, in which stage do you find the bivalent formation? What is your answer, students? In which stage the bivalent formation happens? Tell me. Which stage bivalent formation happens? Quickly in the chat. Which stage bivalent formation happens? It happens in your zygotine. But when it is clearly visible, it is more clearly visible in your packaging. So formation is in zygotine. It is more clearly visible in packaging. Okay. Now, understand what is happening here. Let's take, I have two different homologous chromosomes together. They are called as sister chromatids. These are two are called as sister. But one from this violet, one from this yellow, this one and this one, they are called as non-sister. For example, one from here and one from here. They are called as non-sister. Yes, yellow, everyone, yellow. Right? Non-sister chromatids. Okay. Now what is happening here? Now there is something called as crossing over. Definition, you understand the crossing over. Definition of crossing over. Crossing over is nothing but exchange of genetic material. Exchange of genetic material between non-sister chromatids of homologous chromosome. That is crossing over. Exchange of genetic material between two homologous chromosomes. Non-sister chromatids. That is called as crossing over. Okay. So crossing over is happening. At the point of crossing over, there is something called as recombination nodule formation. Recombination nodule formation is happening. Okay. And there is an enzyme involved here that is called as recombinase enzyme. Okay. Now, crossing over is happening. Once crossing over is over, you can see the yellow part has come here and this pink part has come here, or violet, whatever you call it. Okay. That is called as crossing over. Now, you need to remember here few points. First point is what, students? First point is here. First point is right here. First point is right here, students. That is, bivalent becomes more clearly visible. First point. Now, what is second point? Second point is crossing over. Crossing over is second point. What is third point? Third point is going to be your recombinase enzyme. Recombinase enzyme is what? What is the fourth point? Fourth point is formation of recombination nodule. Formation of recombination nodule during crossing over. And the 4 points, if you remember, entire packeting is in your pocket. It is in your pocket now. Entire packeting is in your pocket right now. Done. Now, what is happening in diplotin? Understand. Very simple. Now the school is over. The school is over. What will happen? Between the two, and the project is also over. The entire project is also over now. Both of you, between the both of you, there was some gum, right? That will disappear. What is happening here is between the two homologous chromosomes, that synaptonemal complex was there, right? The synaptonemal complex will disappear. That is called as dissolution of synaptonemal complex. Point number 1. Second point, recombined homologous chromosomes separate from each other. They are separating from each other except at one point. The two chromosomes are separating except at one point. That one point right here is the X mark. The one point right here is the X mark, and the X mark is called as your chiasmata. The X mark is called as what, students? Chiasmata here. Chiasmata. So if I ask you a question, in which stage of prophase I chiasmata formation happens? What is your answer? The answer should be diplotin. Diplotin, there is dissolution of synaptonemal complex and chiasmata formation. 2 points, that's all. 2 points. 2 points. Now, what happens in diakinesis? One main point or over. In diakinesis, what is happening is the chiasmata. There is something called as termination of chiasmata. The chiasmata will separate out. That's all. Termination of chiasmata, point number 1. Second point is what? Chromosomes condensed fully. Full chromosome formation has happened. Okay. Mitotic spindle assemble here. Nucleolus, everything has disappeared. Disappear. After this entire thing, prophase is over, and then we go to metaphase. All of you know what is metaphase. In metaphase, what we have? Here we have 2 plates. Why 2 plates? One pair of homologous chromosome. One homologous chromosome, second chromosome. See, here we have 2, right? To imagine them this way now. They are like this now. Try to just point it this way now. That's all. See, this is why we see them. Okay. Now that is metaphase. Here metaphase, what is happening? They are formed a metaphasic plate. That's all. Then what happens in anaphase? In anaphase, we pull them apart. Now, is the centromere splitting here? No. Centromere does not split here. No splitting of centromere. Just entire chromosome is pulled apart. Anaphase 1. Then finally, telophase. Okay. So with that, your entire thing is over. Meiosis is over. Done, students? Done? All of you? Quickly in the chat. Any doubt here? Students? Quickly. Now, can we go to the next concept? Students? Next concept. Ready, all of you? Now, the question is, do you want a quick concept from biomolecules, or because in biomolecules there is one concept called as your enzyme concept, which all of you don't understand. So I have the enzyme concept in biomolecules, or do you want anatomy concept? I'll give you option. Don't say both now. Which concept do you want? Do you want a biomolecule concept, or do you want anatomy concept? Anatomy is easy, no? Yes, this 4-day, students, trust me, this 4-day challenge, right? It is so much of background work has gone into it. I have sat around 3-4 hours to get the best content possible for all of you, right? So please like the video. And right, biomolecules or anatomy? Don't say both and all. Both is next level, because I can't teach you plant physiology also, students. Main concept from plant physiology also there. Nikhil Kamath, yes, I know. Why? Students, quickly in the chat, all of you. Okay. A for biomolecules. Okay, B for biomolecules. A for anatomy, B for biomolecules. If I see A more, I will do anatomy. If I see B more, I'll do biomolecules. Go. Quickly in the chat. A or B. Not both. Don't tell both and all. Both and all become next level kashta. Okay, fine. I'll teach you both. What will I do? What will you do without me? Huh? Numerical session was taken down actually. I removed it because none of you watched it. I removed it off. Inky pinky ponky, yeah. Students, I'll do both. It's fine. It's like, I'm asking you A dominant or B dominant. You'll tell me co-dominant. You'll tell me AB. All of you are like that. You like, sir, do co-dominance. AB you take. You like, do both, sir. AB you do. Okay, students, it'll not take more than see, in anatomy, I will do root or stem, any one. I will do root or stem, any one I'll do. Okay, in anatomy. I will not do both. Okay. I'll do anatomy of your root or anatomy of your stem, monocot and dicot. Okay. So what I'll do here is, both are easy. Nothing is difficult. Both are easy. Okay, both are easy. Okay. Now we'll start with something called as enzyme part. Okay. So students, what is a catalyst? Can anyone tell me what is a catalyst? Will the catalyst increase the rate of reaction or decrease the rate of reaction? Quickly, students. Will the catalyst increase the rate of reaction or decrease the rate of reaction? Students, again I'm telling you, this 4-day series will depend on your dedication. If you're not taking advantage of it, it is not my loss, it is your loss. Okay. Quickly, in the chat. Students, I want quick answers in my class at least. Quickly. Quickly, students, tell me in the chat. It will increase or decrease? Students, remember, we always say catalyst as a substance which can alter the rate of reaction. Which can alter the rate of reaction. It can increase or decrease. Increase or decrease rate of reaction. We have inorganic, we have organic. Inorganic, platinum is an example, very good example for nitric acid, right? Synthesis and all. Organic is called as enzymes. So can I technically tell enzymes are nothing but biocatalysts? How many of you agree with me? How many of you agree with students? Enzymes are biocatalysts. Enzymes are biocatalysts. Okay. Now they are biocatalysts. They are organic in nature. All enzymes are proteins except all enzymes are protein except the ribozyme. Ribozyme is a RNA enzyme. Ribozyme is a RNA enzyme, students. RNA enzyme. Okay. Highly specific in action. Okay. Now, what is an active site? Understand what is an active site, you understand first of all. For example, if this is a protein, tertiary structure of a protein. If this is a tertiary structure of protein which has multiple infoldings of beta platelet and alpha helix mixes there. In this tertiary structure, right, there are certain crevices, there are certain gaps, there are certain pocket. I will call them pocket or crevices. In this pocket or crevices, we have a functional group arranged here, and this entire thing is called as a active site. And what exactly happens at active site? To this active site, a particular substrate will come and join, not enzyme. Substrate will come and join to a active site of an enzyme. Will come and join. Okay. Now, how powerful the enzymes are? Small perspective, right? For example, CO2 plus H2O, carbonic acid, 200 molecules of H2O prevailed in 1 hour without a catalyst. But with a catalyst, 6 lakh molecules of H2CO3 in 1 second. 6 lakh molecule in 1 second. Can you believe that? That is the power of your ultimate enzymes. Namaskar I give to enzymes. Okay. Imagine your life without enzymes. There's no life only. Spoiler alert. Okay. Now what exactly happens in an enzyme reaction? Understand. First of all, there is a reactant, second reactant, or substrate. Okay. And before we get a product, right, there is always something called as an intermediate product. Or we always tell in chemistry also, right? In chemistry also we learn that, right? There is something called as intermediate stage. It is there. The thing here is that this intermediate stage right here is also called as transition state. You might have learned this in chemistry. In chemistry you might have learned it. Okay. Transition state. Yes. Now this transition state, right, it has very high energy. Very high energy. Transition state has very high energy. Okay. Now, for example, this is a typical reaction which is happening with the help of enzyme without enzyme. Here is a substrate. Here is the transition state. Very high energy, right? Now, for this substrate to make product, for this substrate to become product, it has to reach the transition state. I'll repeat again, listen to me. For this substrate to become a product, it has to first become a transition state. If it has to become a transition state, it needs some energy. That is where a concept comes, something called as activation energy. Understand? Average energy difference between substrate and transition state. Here, between substrate and transition, there is an energy here, right? This, can you see all of you? Here. This energy here. This is called as activation energy. Here. Activation energy. This part is called as activation energy. Right? Activation energy. So, if you want to reach this activation energy, it is very difficult. Now, I'll give you one small perspective. Understand. I want one volunteer. Students. Quickly in the chat. I want who is the volunteer? Who wants to become a volunteer? Quickly, students. Quickly, I want one volunteer. Let's take Roshni. Let's take Roshni. Okay. Roshni is trying to do something called as high jump. This is our Roshni. We'll give her pink hair. Okay. She's trying to do something called as high jump. Okay. Now what I will do is, this is the high jump barrier. Okay. And here I have sand. Okay. This entire barrier, I will also call it as activation energy. This Roshi, I will call her substrate also. Substrate. Now for this Roshini to run, she'll run. Very difficult to jump here. Tell me students, easy or difficult? Tell me students, easy or difficult to jump? Quickly in the chat, students, tell me. Is it easy to jump over? Is it difficult to jump over? It will tell me, sir, it's very difficult. Unless she is Supergirl or unless she has some jetpack, very difficult to jump. Now, she will call a friend. Bro, listen, I am in a competition of high jump. I am not able to jump. I want you to come and do some scam here. What is a scam? Her friend will come. She'll come silently when no one is watching. She will take the entire barrier, and when no one is looking, she will change the barrier to bottom like this. Where bottom. Friend will come and do scam. Now the barrier is reduced. Activation energy is reduced. Now what will happen? Roshi will come. La 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 la. She'll come and jump easily. There was a decrease in activation energy. Yes. Now, same concept to apply here. Students. This is the activation energy. Transition state present here. Very high energy. Without enzyme, here is the energy. Now, who is the friend here? Who is the friend in need? The friend in need here is the almighty enzyme. Whenever the enzyme is present here, what is happening? The entire activation energy is decreasing, and this much O2 side has to jump now. So substrate will easily cross the barrier. And will become product. That is what is happening. Students, in your entire reaction. How many of you understood, students? How many of you understood? how are enzymes functioning. So what exactly is happening here is, in total we have something called as enzyme substrate plus enzyme complex. This is becoming enzyme plus substrate. It will become enzyme substrate complex. It will become, okay. Then it will become enzyme product complex, and eventually it will become enzyme and product will be separate. This is your entire reaction. Understood, all of you? Understood? Tell me yes or no. Yapa, how much drama I have to do to teach all of you now? All of you understood or not? Tell me in the chat quickly. Students, where is the 300 likes? Quickly smash the like button. 300 is the aim. Okay. Now, can we do anatomy now, students? Can we do anatomy? Now, students, in anatomy, do you want stem or root? Not both. Don't tell both and all now. Do you want stem or root? Quickly in the chat, all of you. Dicot stem, monocot stem, or dicot or monocot root? Quickly. 500 likes target. Who is putting 500 likes target? That is very high bounty number. We don't achieve that here. Students don't like only. They watch, they go, run away. All of you drink some water also. Students, can I ask all of you one thing? Small personal question. The personal question is that, how is your preparation going on? I want everyone to rate your preparation out of 10. Go, students, quickly. I can see a lot of STEM, so I'll do STEM. Students, quickly rate your I'm going to say training or program. Shivraj, I will talk to you separately. I can't tell you the solution here. Okay. Try to text me on Instagram, I'll call you. Text me on Instagram, I'll give you a call and we will talk. Don't worry. You text me on Instagram, I'll give you a call and we'll talk and call. I'll talk to you, don't worry. 5, 4, 6, 2, 1. Hey, Ravindrani, why 6? Okay. That is done. So let's go with the next concept today. I think, students, we have a lot of concepts left still. So before we go further to anatomy, there's a small thing which you need to understand. Okay. Let me teach you one small concept right now. Small concept. Okay. What is a small concept to understand? Okay. In vascular tissue system, there are two types of arrangement of vascular bundles. There is something called as your radial and conjoint. Understand the difference. Okay. Understand, students, quickly. What is the meaning of radial? In radial, what is happening is xylem and phloem are present in its own radii alternatingly. They are very shy to each other. They will not hold hands here. Very shy. Okay. Bad touch. Okay. They're present separately here. Separately. Xylem phloem are separate here. Okay. Now understand what exactly happens in conjoint. In conjoint, xylem and phloem are present in same radii. Xylem phloem in same radii they are present. In same radii they are present. Okay. So this is present in the case of root. This is present in the case of stem. Roots and stem. Clear? Okay. Now, under conjoint, again there are two types, bicollateral and collateral. Understand? In bicollateral we have phloem. In collateral we have phloem. And here you need to focus here is that, students, the xylem is towards the pith here. Xylem is present towards the pith, towards the center, xylem. Okay. Now collateral can be of two types again. In the case of dicot stem, right? Mainly what happens is, between phloem and xylem, or xylem and phloem, there is a one more layer called as cambium, and this cambium is responsible for secondary growth. And this cambium is completely absent in the case of monocot and also leaf. Why? No secondary growth. There. Okay, no secondary growth. Hello, Gopika ma'am is here. Namaskar to Gopika ma'am also, all of you. Namaskar to Gopika ma'am. Gopika, students, trust me, I'll tell you honestly, okay. I have never seen such dedicated teacher as Gopika ma'am. I have seen how she is suffering right now. Ma'am is barely able to talk. Okay, ma'am is barely able to talk, and she's taking a session for all of you on 3rd. Okay, so I want all of you to quickly go to the session and smash the like button there first of all right now, right? And please attend this class, because if someone is coming out of pain, coming out of, for students so much, you should show respect. Tell me yes or no? All of you. Tell me yes or no? Right? Show the respect to ma'am, because I have seen how ma'am is suffering right now, how much of pain she's going through, right? If ma'am is teaching you after all that, you have to be very grateful for such teachers. Okay, be very grateful, all of you. Now, whenever there is cambium present between xylem and phloem, right, that is called as open type. Okay, whenever there is no cambium, for example, in the case of your leaf, right, such a condition is called as closed condition. So open condition, there is a cambium. Closed condition, cambium is not present. I don't think you need to even find it difficult, right? No. Phylloderm is not in your syllabus, or simply will waste time upon that otherwise. Okay, so it is not there for you. Don't worry. Secondary growth is removed. Okay. Now, students, we will do stem. All of you ready? Students? Ready for dicot stem? Students, if you want these notes, Telegram channel is going to be their notes. Okay, and you will get all the notes. More, I will include even root notes for all of you. What is collateral crazy? Understand collateral, very simple. I'll show you the diagram again. You will understand better. What exactly happens in collateral? See this. In the case of collateral, what is happening is, it is similar to conjoint. You might be thinking, sir, conjoint and collateral, are they same to same? No, there can be a difference. In conjoint, both are present together. But in case of conjoint, what can happen is phloem can be present towards the pith. If this is the pith here, phloem can be present towards the pith in conjoint. But in the case of collateral, xylem is present towards the pith. In the case of collateral, xylem is present towards the pith here. Clear, all of you? Clear, students? Xylem is present towards the pith here. That's called as collateral. Now we go to dicot stem. Students, ready? Dicot stem, ready all of you? Quickly in the chat, all of you, tell me in the chat. Ready for dicot stem? Ready all of you? Dicot stem. Nothing, students, very easy dicot stem. I'll show you, I'll tell you exactly what you need to read here. It's a very, very easy concept. Okay. And if you download the notes, practice yourself once, it becomes even more easier. Okay. Can we start? Listen to me, all of you. I'll tell you the entire functions, layer by layer, I'll tell you the functions. Okay. In the entire dicot stem, the outermost layer is called as epidermis. Outermost layer, protection, single layer of epidermis. It has something called as trichomes. Students, can anyone tell me what is a trichome? Trichome is nothing but it is called as stem hair, right? And trichome, as a matter of fact, can also secrete certain substances, secretory also in nature. Okay. Then there is no chloroplast, obviously, because here epidermis, outermost layer, right? It has intercellular space is completely absent here. Cuticle may be present, may be absent. Stomata may be present, may be absent. So that is your entire epidermis. The only thing which you need to remember here is single layer, protection. This is easy to remember. No. Second layer, this entire thing is called as your ground tissue system. Entire thing is called as what? Ground tissue system. It has 3 layers here. The entire ground tissue system has 3 layers. The first layer is called as hypodermis. The one right below it. Can you see here? It says collenchyma here. That is your hypodermis. Then we have cortical layer, and the innermost layer is called as endodermis. So here we have 3 layers: hypodermis, cortical layer, and endodermis. Endodermis right here. Now, what is the function of each one of them? Understand. Hypodermis is made up of collenchyma. You have to remember that. Okay. ICS is absent, intercellular space is absent here. Few layer, mechanical support, photosynthetic in nature. If you know collenchyma, you can write all this very easily. Now, cortical layer, parenchyma, round thin wall cells, ICS is present here. It's parenchyma packaging. The innermost layer is called as endodermis here. Here, endodermis has something called as starch grain in the form of starch sheet. Very, very important. So remember students, every single time we talk about endodermis in the case of dicot stem, it should come to your mind. This should come to your mind, students. Okay? Dicot stem. No. Next layer. Then we have something called as pericycle. Now, how many of you like watching moon? Because I like watching moon. Moon is very nice. Okay. Full moon, half moon. Full moon is lunar. Full moon. Half moon is called as semi-lunar. Semi means half. Semicircle. Right? So, half moon. Now, still look at the shape here. Half moon shape. Okay. So, pericycle is semi-lunar in shape. Semi-lunar in shape. Okay. It is wavy. Can you see? It is wavy here. Wavy. Not baby, wavy. Okay. Wavy here. Okay. And it is made up of sclerenchyma. Very important point. Sclerenchyma. Okay. Sclerenchyma. That is a pericycle here. Pericycle. Now, innermost layer. After pericycle, what do we have? We have the entire vascular bundles and the pith here. And the pith here. Okay. We have the entire vascular bundles here. Can all of you see the vascular bundle, students? How does it look like? Does it look like a ring arrangement or not? Tell me, students. If you look at the vascular bundles here, it's the ring arrangement. Yes, sir. Ring arrangement we can see. Are they conjoined in collateral? Yes, they're conjoined in collateral. How come, sir? See, xylem is towards the pith here. So they are conjoined and collateral. Phloem is away from the pith. Okay. Clear? We have endark. We can find endark type of system here. Endark. Okay. Endark. You can find here. Apart from the students, it is also open or closed. Students, can anyone tell me? Is it open or closed? Quickly in the chat, students. Is it open or closed? Yes, we are under the same moon. Open or closed? Quickly in the chat. Tell me. Look at the diagram and tell me. Look at the diagram and tell me, students. Is it open or closed? Quickly. It is going to be open. Why? Because of the cambium is present here. Last part is called the pith. Pith is very large. ICS is present, made up of parenchyma, and that's it. But if you look at a monocot stem, dicot stem, pith, you should know. Vascular bundles, oh, it's given here. Ring arrangement, endark, conjoined, collateral, open. Pericycle, ground system, tissue system, ground tissue system, epidermis. 1, 2, 3, 4, 5 slides. Just 6 slides, if you remember, entire dicot stem is over. 6 slides, few points you can remember. Now, monocot stem is not very different, but there is a major difference if you look at it from the outside. There, in the case of dicot, we had a ring arrangement. But in the case of monocot, the vascular bundles are completely scattered here. So, if you get a question examination, scattered vascular bundles, immediately, without thinking, you should write monocot stem. Okay, monocot stem directly. Okay, epidermis is cuticle, stomata is present. Hypodermis is 2 to 3 layers, sclerenchyma here. Hypodermis is present here. Remember, hypodermis is present. Okay, here sclerenchyma. Why? Because collenchyma is absent here. Collenchyma is present only in the case of stem, dicot, not in monocot. Monocot, collenchyma is absent. So this becomes very important line here. They can ask you a question around it. Question around it. No. Ground tissue is not differentiated. It's made of parenchyma. Here, if you look at all the vascular bundles, on the outside we have many small size. In the middle we have large size, very few. Outside many small size, inside very less, size is more. Vascular bundles are conjoined, collateral, and closed here. Closed why? Because it is monocot. Monocot is closed. Here, entire vascular bundles are covered by a bundle sheet that is called as sclerenchyma, and this also is a PYQ, students. Phloem parenchyma is absent here. And if you look at the entire structures here, you can see a small round structures inside the vascular bundles, that is called as water cavities. Here we can also find water cavities here. We can find water cavities on the inside. Here, tiny water cavities. Okay. Students, with that your entire anatomy is also over. Entire anatomy is also over. Now, can we come to the big boss of your class 11 topics? Can we start, students? The entire plant physiology. Now here I'll be mainly focusing on few things, not the entire plant physiology, right? I'll not be focusing on the plant growth regulators. Plant growth, easy to go, right? We'll be mainly focusing on photosynthesis in higher plants as well as respiration in plants here. Again, not all topics, only few main topics where it is difficult to understand and questions come from. Can we start? Can we start, students? Can we start? First we'll do photosynthesis in higher plants, then we'll do respiration in plants. Students, quickly smash the like button, all of you. Can anyone remind how many likes do we need more? How many likes do we need more, students? How many likes do we need more? This is the big boss only. I can't see the like, students here. I forgot to see. 1 second. 274, right? Hardly few. Hardly few. Okay. So let's start by a simple concept of your chloroplast. Because if you don't understand this right now, forget the chapter. Forget the chapter. Okay. Because this is the basics which you need to know this chapter of photosynthesis in higher plants. If you don't really understand the structure of chloroplast, what exactly is present where, chapter gone. Understand. This is the structure of chloroplast here. Right. Chloroplast is made up of two layers, outer layer, inner layer, two layers here. Now inside the chloroplast, there are certain coin-like structures called as thylakoid. All of you can see. There are many coin-like structures called as thylakoids here, thylakoids. This entire stack of coins is called as grana. So each one is called as a thylakoid. Entire stack is called as grana. Between the two thylakoids, the connecting link is called as stromal lamella. Two things, the connecting link, stromal lamella, right? And it has a matrix around it. The matrix is called as stroma. Inside the stroma, dark reaction happens, and in the thylakoid grana, light reaction happens. Even the stromal lamellae, light reaction happens. Light reaction happens. So the first thing which you need to know here is, where is dark reaction happening? Where is light reaction happening? Clear? Dark reaction is happening in the stroma, light reaction is happening in the grana thylakoid and even stromal lamellae. Clear? Now the next thing which you need to know will be the foundation for cyclic and non-cyclic photophosphorylation. Okay, basics. Understand. Here on the thylakoid, we can find both PS1 and PS2, both are found in thylakoid. While on the granule, on the stromal lamellae, we only find PS1. Only PS1, both are present here. Both are present here. Clear, students? Clear? Clear, students? Here we have PS1, here we have both. Okay. If that is clear to you, students, you need to understand in your entire photosynthesis, right? There are two main things, light and dark reaction. What exactly happens in light reaction? In light reaction, there is trapping of light. Dark reaction, food preparation. Food preparation. Imagine light reaction as you're working really hard, earning the money. You're earning the money in light reaction, right? And later on you go to a nice restaurant, buy the food and eat. The buying the food and eating or making the food and eating is called as dark reaction. Earning the money is called as light reaction. Clear? And one more thing I'll tell you, students, light reaction, dark reaction, both can happen in daytime. Both. Hey, don't ever think that dark reaction will happen in night time secretly when no one is looking. No. When the entire world is sleeping, plants in night time, they will do dark reaction. No. Dark reaction can also happen in the daytime. It is just that dark reaction does not require light. Does not require light. That's all. Does not require light. Okay. Now, in the light reaction, we have something called as non-cyclic and cyclic photophosphorylation. Okay. Now, what is the meaning of photophosphorylation? The meaning of photophosphorylation is that we are using light energy and making ATP. Phosphorylation is formation of ATP. So we are using light energy, exciting electrons, using that energy and making ATP. Okay, making ATP. Here is a non-cyclic photophosphorylation. Non-cyclic. Here is a non-cyclic, students. Here is a non-cyclic. Non-cyclic. As you can see here both PS2, PS1 both are involved here. In PS2 we have, it works at 680 nanometer. PS1 700 nanometer. Very important. Now in 680 what is happening? When the photons are coming, light is coming, electrons are excited. And the primary electron acceptor in the case of your non-cyclic, it's going to be pheophytin. From pheophytin, it's going to PQ, CY, PC. What is PQ? Pheoplastoquinone. CY, cytochrome B6F complex. PC, plastocyanin. So as the electrons are passing, the energy is used to make a ATP here. ATP is made here. Now finally the electrons will go to now PS1. Now again from PS1 electrons are excited. Again it is taken by primary electron acceptor, that is FES. Iron sulfur centers. FES. From FES it goes to FD, ferredoxin, where the final electron acceptor is going to be NAD+, which undergoes reduction to produce NADPH. Now what is reduction? Reduction is nothing but gain of electrons or gain of protons. That is called as reduction. So it is gaining electrons and gaining protons from the stroma, and with the help of enzyme FNR, ferredoxin reductase, right? NADPH reductase enzyme, reduction is happening. And this entire process, there is a continuous supply of electrons. The continuous supply of electrons is coming from the photolysis of water. That is, breakdown of water is happening. Splitting of water is happening. So water is being split in the lumen of thylakoid. What is lumen of thylakoid? For example, if this is a thylakoid, right, all of these reactions are happening here. Here, let's say here we have PS2, here I have PS1, right? PS2, PS1 is present. All the splitting of water is happening inside. This is called lumen. Inside the lumen, splitting is happening. All the reactions happening here. Reaction is happening here on the membrane. So this entire reaction is happening on thylakoid membrane. Thylakoid membrane. Okay. Now understand all of you. So here water is split up. Electrons are taken here. Hydrogen is released for your ATP synthesis, and here oxygen is released as a byproduct. So it goes on. Now students, can I tell in the chat, all of you, in this entire reaction, students, is there splitting of water? Yes, sir. Splitting of water. Is there NADPH formation? Yes, there is NADPH formation. Is there ATP synthesis? Yes, there is ATP synthesis happening here. Oxygen is released? Yes, O2 is released. Splitting of water, students. Splitting of water. Splitting of water. O2 is released. O2 is released here. Here electrons are going in a Z scheme, students. Can you see? It's like a Z scheme. Is it cyclic or non-cyclic? It is non-cyclic. Why non-cyclic? Because electrons are not coming back. It is non-cyclic because electrons are not coming back. It is going away. Non-cyclic. Okay. Now what about cyclic? In the case of cyclic, we have only PS1. Now again think back. In the chloroplast, there was a region where we have only PS1. That was stromal lamellae. So this is happening on the stromal lamellae where we find only PS1. Cyclic is happening. In cyclic, electrons are excited again to primary electron acceptor, FES, FD, PQ, CY, PC, electrons set back again. Again, ATP synthesis happening. But here, students, is a splitting of water? No splitting of water. No oxygen is being released. There is no NADPH formation here. No NADPH formation. There is only ATP formation happening here. Only ATP formation. And that is your cyclic, student. That is your cyclic. That is going to be your entire cyclic photophosphorylation. Cyclic. Okay. Now, here is your Krebs cycle. Here is your Krebs cycle, also called as your C3 cycle. Also, not Krebs cycle, student. Kelvin cycle. Here is the Kelvin cycle, also called as your C3 cycle, also called as PCR cycle. photosynthetic carbon reductase cycle, reduction cycle. Okay. Now here what is happening here? I'll teach you in a very simple manner. It's called as 3-2-1-0 method. What is the method here? 3-2-1-0 means 3 means 3 steps. 3 steps. Carboxylation, reduction, regeneration. Again, 3 means 3 ATP being consumed. 2 ATP here, 1 ATP here. Okay. Now what is 1? 1 means, what is the 2 here? 2 is nothing but 2 NADH. 2 NADH is being used here. What is 1? 1 means 1 CO2, this entire cycle is happening. 0, no energy is being released here. Now understand the concept. 3 steps. First step, what is happening? RUBP, also called as ribulose 1,5 bisphosphate, is undergoing carboxylation to produce PGA. What is PGA? Phosphoglycerate. Now here we have 5 carbon, 1 carbon, 3 carbon. Why? Very simple. 5 carbon, 1 carbon, 3 carbon into 2. What is 3 into 2? 6 carbon. So we get 2 molecules of 3 PGA here. 2 molecules of 3 PGA. Okay. So 6 carbon maintained. Now this second step, it undergoes reduction. In this reduction step, there is 2 ATP formation, so 2 ATP being used and 2 NADH used. Now can anyone tell me in the chat, anyone, where is this 2 ATP and where is this 2 NADH coming from? Anyone? This 2 ATP and this 2 NADH is actually coming from your light reaction. Light reaction is coming from. Light reaction is coming from. It is being used here. Okay. Now we get triose phosphate. This triose phosphate later forms glucose. 3 carbon maintained. It undergoes regeneration. Again, 1 ATP is used here to make RUBP again back. So in total there are 3 ATPs consumed, 2 NADH is consumed for 1 CO2. Now if they ask you a question in examination, what about for one glucose, sir? Glucose. For example, if they ask you C6H12O6, how many energy or how much energy is used for one glucose? You will tell me, sir, here we have 6 carbon. 1 CO2 has 1 carbon. For 1 carbon, we need this much. For 6 carbon, how much? Just do into 6. Into 6. 18 ATP, 12 NADH. That's all. That's all for your entire glucose. For entire glucose, 18 ATP and 12 NADH. So, here is your C4 cycle. Now, what is the difference between C3 and C4? The main difference is that in your C4 cycle, there is no photorespiration. What is photorespiration? The photorespiration is nothing but, students. Here we see an enzyme called as Rubisco enzyme, right? This Rubisco enzyme is very moody. Very moody. Is okay. Here it is forming a bond with CO2 in this reaction. But sometimes what this Rubisco does it forms a bond with oxygen to undergo C2 cycle. That is called as photorespiration based process. Okay. But in the case of your C4 plants, it is not happening. In the case of C4 plants, it is never happening. Okay. Now understand. Here in C4 cycle, CO2 is received by PEP. Here it was RUBP was receiving here. Here we have PEP. What is PEP? Phosphoenol pyruvate. When 3 carbon plus 1 carbon, we get a 4 carbon compound called as OA, oxaloacetic acid. That is why it is called as C4 cycle, 4 carbon. Here we had 3 carbon molecule, C3 cycle. I hope it makes sense now. Okay. Now this OA, oxaloacetic acid, changes into malic acid. Malic acid transported here. Malic acid gets converted to OA again. OA is 4 carbon, gets converted to pyruvic acid is 3 carbon. 4 carbon, 3 carbon, 1 carbon is missing, that is CO2. This CO2 will enter the Krebs cycle and enter the Calvin cycle here again, Calvin cycle. Again, Rubisco is present here. Here we have enzyme called as pepcase enzyme here in this reaction. So pepcase on the top in mesophyll cell. In the bottom in the bundle sheath cell, we have the Rubisco enzyme. Pyruvic acid again goes back up, converts back to PEP. So what question they will ask you here? The question may ask you is, in which cell we find Rubisco? Answer is bundle sheet. They can ask you, in which cell do we find pepcase? Mesophyll cell. In which cell does the Calvin cycle occur? Calvin cycle occur in your bundle sheet. In which cell initial fixation of CO2 is happening? Mesophyll, initial fixation of CO2. Those are the main questions which they can ask you repeatedly for C4 cycle. C4 cycle. Or they can use diagram. Can you see? Transportation was happening here. Decarboxylation happened here. Decarboxylation happened here. And transport happened again. Regeneration happening here. Regeneration. That's all. Now students, we come to the last chapter for today. Now we come to the last chapter, that is called as your entire respiration in plants. Can we start, students? Can we start? Can we start? Last chapter for today. Can we start, students? Can we start? Here I will teach you only Krebs cycle because that is only important here. Glycolysis is very easy. Glycolysis is going to be very easy, right? So I'll teach you only Krebs cycle here in the best way possible. Can we start, students? Can we start? 300 likes done. Wow. Proud of all of you. Proud of all of you. Can we start, students? Can we start? Students, are you ready for part 2? In part 2, same way I will teach you the entire class 12th. How many of you ready for part 2? Let me ask you before I start this. How many of you ready for part 2, students? Same way I'll teach you for part 2. How many of you ready for part 2? Ready, ready, ready? ETS is very easy, students. ETS is very easy. See, in part 2, I will be teaching you main focus is going to be reproduction, genetics, biotech. I don't think so. I'll do biotech. Biotech is very easy. Biotech, ma'am will do biotech. Ecology, because I feel some of you are ignoring ecology, right? If you remember, students, last year NEET examination 2024, from biodiversity and conservation, we had 5 questions or 4 questions, if I don't remember, right? 4 to 5 questions we came from biodiversity, right? There were 1 question, 2 questions from ecosystem, 2 questions from your organism and population. So if you're ignoring your ecology, you are easily losing your government seat. Okay, so do not take entire ecology is granted. I know it is very easy, you keep it to the last moment, plus don't do that. Okay, so attend the part 2 where I'll be teaching you the entire ecology, most points. Okay, because my main focus is going to be ecology there, ecology as well as your reproduction. Okay, so students, with that we come to the last topic today. Before I teach you the entire Krebs cycle, right, you need to understand a small concept called as oxidative decarboxylation. Understand. Oxidative decarboxylation is a reaction which is happening called as a link reaction and also called as gateway reaction. What exactly is happening here? Understand. Okay. First of all, we have something called as pyruvate from your Krebs cycle, from your glycolysis. Okay, pyruvate from your glycolysis. What is happening here is initially there is a removal of CO2. Decarboxylation, removal of CO2. Now, once CO2 has been removed, the remaining part is called as acetyl group. Okay, acetyl group. Remaining part. If you remove CO2 from here, we are left with COCH3. That is called as acetyl group. Now, this acetyl group undergoes oxidation. Now, what is the meaning of oxidation? The meaning of oxidation is loss of electrons or loss of protons. So here we have what? Electrons and protons are lost. Now these electrons and protons are taken up by your NAD+. Now what do you call, students? If NAD+ is taking electrons and taking protons, what do you call that? Reduction. So this NAD+ undergoes reduction to produce NADH. And that is called reduction. Okay, and then we add the coenzyme and we get the acetyl CoA here. So that is called as oxidative decarboxylation. That is, there is oxidation happening here, decarboxylation happening here, and this same reaction also happens in your Krebs cycle also. Okay, Krebs cycle also. See this, all of you. This is a simple Krebs cycle which I have given you. My favorite Krebs cycle, which I always teach. Why is it called as Krebs cycle? It's called as Krebs cycle because the inventor, right? Hans Krebs, inventor. So who discovered, not invented, discover. Because Krebs cycle is always is discovered, not invented, discover, right? C4 cycle is also called as Hatch and Slack pathway. Remember that. C4 is also called as Hatch and Slack pathway. Karmani, I will do ecology. Don't worry. Okay. Now, understand your Krebs cycle here. Okay. Now, what is the first step? First step is called as citrates. Citrate and isocitrate between we have something called as oxaloacetate. Now, to this oxaloacetate, acetyl CoA will come and join. 4 carbon, 2 carbon, 4 plus 1 is 6 carbon citrate. Citrate becomes isocitrate, then alpha ketoglutarate. Now here to here is the first OD. What is OD? Oxidative decarboxylation. Between your isocitrate and alpha ketoglutarate, we have the first oxidative decarboxylation. What is happening here is this isocitrate, here one CO2 is removed. Then it undergoes oxidation. When it undergoes oxidation, the electrons and protons are taken up by your NAD+, and that's how we get NADH here. This NAD+ will take up all the electrons and protons, and it undergoes reduction to produce NAD+. Same thing which happened here, students. Same thing which is happening here. NAD+ is taking the electrons and protons from your acetyl CoA, forming NADH. Same here, isocitrate is undergoing oxidation. It is releasing electrons and protons that is taken up by your NAD+. That's why we get NADH here. And that's why we get here CO2 also. CO2. Oxide decarboxylation. Now again, alpha ketoglutarate to succinyl coenzyme A, we have second OD. Not overdose, oxide decarboxylation, second one. Okay. Then succinyl coenzyme A to succinate, we get one GTP. And listen to me very carefully. This GTP right here is the only substrate level phosphorylation. The only substrate level one. Okay. Then we have fumarate, FADH, malate, one water, one more NADH. Now there's a small trick here. Trick here is going to be, officer, can I keep selling sandwiches for money? O in the officer stands for oxaloacetate, C for citrate, I for isocitrate, K for alpha ketoglutarate, S for succinyl coenzyme A, S for succinate, F for fumarate, M for malate. With that small trick, you can remember. The only thing which you need to remember here is that wherever OD is happening, wherever we get fumarate, and when are we getting GTP. If you remember that, that's all is Krebs cycle. That's all is Krebs cycle, students. That's all Krebs cycle. Okay. Clear, students? Clear, all of you? Clear or not? Quickly in the chat, tell me. The only thing which you need to remember here is this part. Mnemonics, wherever we have between isocitrate and alpha, first OD, alpha ketoglutarate to succinyl coenzyme is second OD. That's all you have to remember. Nothing more. And always remember, students, whenever there is OD, there is release of CO2, release of NADH. Here we have, here and here, we have normal only oxidation. We have only oxidation here, nothing more. That's why there is no decarboxylation here. There is no decarboxylation here, students. Okay. And with that, we come to the end of today's lecture, students. How was it, students? Can I get a normal feedback today? Can I get a proper feedback in the comment section? Should I include more topics next time? Should I decrease the topics? What is going to be the feedback? Let me know in the comment section. Okay. How was today's class? Did you enjoy? Did you learn a lot? Did the concepts become much more clear? Were you able to, understand your entire fungi? Able to understand the cilia and flagella? Then we went on to your meiosis. Then we did biomolecules. Then we did your anatomy. And then we came to plant physiology. Right? We did your entire light reaction, dark reaction, and we did your Krebs cycle. Okay. So if you want more topics, PGR, nothing is there, students. PGR is like, you have to remember. I'll give you PDF, you remember that, you're good to go. Nothing is in PGR. Okay. PGR is so easy. Okay. So students, if you want more concepts, then this session, let me know in the comment section, right? I will bring to you, right? Now students, again, this is just overview, overall revision happening here, right? If you're not aware, the crash course has started already. In crash course, some of you have joined, so many of you have joined, right? In crash course, we are doing detailed revision. In crash course, we're doing detailed revision. Recently, I finished the entire cell chapter in just 2 classes. Just 2 classes, entire cell chapter. I'll be finishing the entire cell cycle chapter in 1 class. I'll be finishing the entire plant kingdom in 2 classes. So that's how I'm doing in my paid batches. So if you're not joined till now, this is your clue. Okay. So where can I get a full NEET syllabus? Ananya, I will send in my Telegram today. If you want the PDF, I'll send in Telegram today the entire PDF of your syllabus one more time. Okay, don't worry. Okay. So thank you so much, students, for today's class. And can I scoot? Students, can I scoot now? All of you. Or do you want me to ask you some few questions? Do I want me to ask you some few questions? Okay. I'll ask you 10 questions. Let's see who can answer. I'll ask you just 10 questions I'll ask you. Okay. Tell me which class of fungi is called as fungi imperfecta. Quickly. Which class of fungi is called as fungi imperfecta? Quickly. Fungi imperfecta. Which class of fungi is called as fungi imperfecta? Quickly in the chat. Answer in the chat. Tell me, students, in which of the 2 following fungi classes we get fruiting bodies? We get fruiting bodies. Quickly. In which of the 2 following fungi classes we get fruiting bodies? Ascocarp and Basidiocarp. Where do we get Ascocarp and Basidiocarp? Quickly. In which class of fungi we do not find asexual spores? In which class of fungi do we not find asexual spores are completely absent? Quickly. Tell me, students, what is the array of arrangement in your centriole? Array of arrangement in centriole, quickly. In centriole, what do you find? 9 plus 2 or 9 plus 0? Centriole. 9 plus 2 or 9 plus 0? Quickly. Next question. Cilia and flagella are made up of which type of cytoskeleton? Cilia and flagella are made up of which type of cytoskeleton? Quickly. Next question. Which is the phospholipid present in the lipid bilayer? Which is the phospholipid present in the plasma membrane or lipid bilayer? Next question. Next question. Okay. Name the scientist who gave the plasma membrane? Who gave the fluid mosaic model and the year also? Who gave the fluid mosaic model and the year? Okay. Next question. Name a metal ion cofactor. Example of metal ion cofactor. Next question. Name the enzyme involved in conversion of sucrose into glucose. Sucrose into glucose. Name the enzyme present in the bundle sheet of C4 cycle. Next question. Which is the primary electron acceptor in your cyclic photosynthesis? phosphorylation. Primary electron acceptor. Primary electron acceptor in your cyclic photophosphorylation. Quickly. Next question. Next question. Tell me the hormone responsible for internodal elongation in deep water rice plants. Tell me the hormone involved in internodal elongation in deep water rice plants. Next question. Name the hormone responsible for seed germination. Seed germination. Ravindrani, wrong. It is ethylene. I told internode elongation, deep water rice plants. It is going to be ethylene, not gibberellin. It is ethylene, not gibberellin. Deep water rice plants. Don't make mistakes like that. Name the hormone responsible for root hair elongation. Name the hormone responsible for root hair elongation. Students, do you realize I am choosing the NEET PYQs in my head and I'm asking you orally from nowhere. I'm not looking here and there. All the questions are in my head. Name the hormone responsible for root hair elongation. Quickly. If anyone writes auxin 1, I'll throw this pen at you. If you tell auxin, root hair elongation is ethylene again. Root elongation is auxin. Okay. So students, I think I'll stop here. Students, I think I'll stop here. If I keep asking, it'll go on for 1 more hour, right? I can keep on asking questions from entire class 11th, entire class 12th. Okay. So I think I'll stop here, students. Thank you so much. Hopefully all of you enjoyed the class. Hopefully the brain stimulated now. Okay. So I'll meet you in the day 3. Okay. I'll meet all of you in day 3. Again, I'll meet all of you in day 3, 10 a.m. Okay. So join Telegram channel for all the notes. Notes will be given only in Telegram. So join Telegram for notes. Okay. So thank you so much, students. Bye bye. Take care, all of you.